Yeah, so this is a very excited by this presentation. It's an oral presentation today by a PhD student, Chen Zhu. So we have a collaboration with Penn and MIT and University of Miami and ORNL, where we try to tackle this complex data in histopathology. Histopathology is the biopsy that determines the diagnosis of a patient. Every cancer has to start with the histopathology diagnosis for the most part, except for, there are exceptions to that. But when these glass slides, literally, the traditional approach is to look under the microscope and for the pathologist to give a read. Certain solid tumors, lymphomas, have complex microenvironment, complex layers, and sometimes not discernible for the human eye. And the experienced pathologists would have a great idea of what's going on, but are not able to articulate it. One of the downfalls of this approach is it's a very descriptive, non-quantifiable approach in histopathology. So what we have attempted to do in lymphoma, which is another challenge, is to try and digitize those images of pathology in the same way that the pathologists look under the microscope. So these are Z-stacked images that you can zoom in and zoom out. So you put them on the screen, and you can actually, it's a digital microscopy. And using that approach of low resolution, high resolution, we're able to reconstruct the computer vision extracted information. And the computer vision is a term for what the algorithms and the deep learning and the machine learning do. And it's a very rapidly advancing field in AI, if you will. So what we have actually used is taken those images down into a code environment like Python, wrote our own codes, and built upon the other open source codes that are available, and we're able to create a pipeline that allows this image analysis. So we took data sets from the public domain that has the Z-stack image and very rich data, because these are complex images, gigapixel images. If you take a photograph, it's a single two-dimensional view, but histopathology has not just the zoom effect, but also has the complexity of the cells in the microenvironment. So we took the data sets, one from the TCG, the Cancer Genome Atlas, the other from Stanford, which is a published data set. We put them together, we cleaned up the data, we loaded the images, so about 225 images, and then we trained algorithms. Historically there are traditional algorithms that use what's called as deep learning or neural network. And there's something called convolutional neural network, which works sort of like how we see and we learn and we discern things. So the eye sees an image and the brain sort of labels that. Similarly, the neural networks do the same thing in this algorithm. So we used a more contemporary deep learning process called self-supervised learning, which means that you don't really need to have a label for the entities. It depends on where they are in the position. We're able to pull together all the data, and we create what's called as a vector and a position, and these are we then make all the image information that is obtained as a data point, and we then compute. So just to give you a scale, if in this particular room we have all kinds of equipments, and there are about 10, 20, right? So the scale that I'm talking about in a single glass slide is about 30 million. So you need a computer to be able to actually sort everything out and also give you meaningful information. So we use the self-supervised training, and by the way, the self-supervised training is also something that's very popularly known to everybody. That's ChatGPT. It uses a very similar transformer-based training. So we took the self-training approach, and we found that it was able to give a lot of information at the level of the slides at low resolution and high resolution, and also at the cellular level, more importantly. And the cellular level gives you the context of the cell, the size of the cell, the shape of the cell, what the surrounding cells are. And so what we are able to do is reconstruct entire spatial information in a glass slide using computer algorithms, and we found the accuracy at the rate of 94 to 96%, we're excited by this because it's a challenge in lymphoma, because your immune cells and the lymphoma cells are very similar. It's very hard to distinguish. You have multiple patterns under the microscope, so the computer vision can potentially augment whatever the pathologist does. Not only that, we can create databases, we can collaborate, and you can get second opinions very easily at the push of a button, and of course following all the HIPAA rules. And more importantly, I think we can use this to integrate this with genomic data, with clinical data. And the other important point is that pathology so far has not been computable. It's been a label. I think with this approach, we bring pathology also into compute-ready environments, so suddenly you have very rich data sets, so your predictions are going to be much better. So this is the presentation.